this opportunity to welcome me back to this other session. And today, all right, now we are going to look at what we call the Engelmanns experiment on action spectrum of photosynthesis. So most teachers don't teach this part, but you people who are listening to me, this is also another experiment on action spectrum. So without wasting time, let me look at a description of the Engelmanns experiment. Here we are saying the filaments of green algae, the Chorodophila of the genus Pseudomonas, are placed in a drop of water on a slide, then illuminated with the light of different wavelength and observed under the microscope. So the word illuminated, that word means just exposing. All these organisms are exposed to what? To light of different wavelength. So the control experiment involves mounting the algae on a slide in water with aerobic bacteria in total darkness, and thereafter exposing the slide to what? To light. Now there they are saying that here they have exposed the control experiment involves mounting the algae on a slide. There you can look at the results of the Engelmanns experiment. They have mounted some algae on the slide. There I'm seeing water there. We have aerobic bacteria. We have the filament of the Chorodophila. Down there you have ultraviolet, which is 350, violet, blue, green, yellow, orange, red, and infrared. We are saying that this is exposed in water with the aerobic bacteria in total darkness, and thereafter exposing the slide to light. So when we talk about aerobic bacteria, those are the bacteria which use what? Which use oxygen. And from there, let us look at observation 1. You are saying the motile aerobic bacteria cluster near the filament in the region of blue light. Near the filament on the region of blue light. Sorry for that. On the region of blue light. Our first observation. We are saying that the motile bacteria cluster near the filament in the region of blue light around 450, around here. There you are seeing this part around here, 450 nanometers here. So you are saying that the motile aerobic bacteria cluster near the filaments in the region of blue light, which is around 450 there, and around 600 what? 650 nanometers. Now that is the observation. Now our deduction 1 is like, since the distribution of aerobic bacteria is in the response to the concentration of oxygen, which is a byproduct of photosynthesis, then red and blue light are the most effectiveness for photosynthesis. There we are saying that these bacteria, the aerobic bacteria, for them they use what we are calling oxygen. So they concentrate more on those areas where photosynthesis takes place, because oxygen is the byproduct of what? Of photosynthesis. Then observation 2, we are saying the motile aerobic bacteria cluster around the edge of the cell adjacent to the what? To the chlorophyll. Adjacent to the chlorophyll. We are saying that the motile aerobic bacteria cluster around the edge of the cells adjacent to the chloroplast. Clustering means they actually, it's like they group together around those adjacent cells to what? The chloroplasts. Then The deduction is that oxygen is more concentrated near the chloroplast, which shows that the chloroplast is the site for photosynthesis. Yeah, that these motile aerobic organisms, the word motile means these organisms can move from one area to what? To another. That's why you are calling that motile. You'll find them that they will cluster around the chloroplasts. Reason being that around those chloroplasts there is much concentration of what? Oxygen. So this proves that chloroplast is the site for what? For photosynthesis. Then our observation 3 is like the aerobic bacteria of the slide previously in the dark are immobile, but later cluster around the algae filament on exposure to what? Exposure. We are saying that those bacteria which are previously in the dark are immobile, means they do not move. Now, when we say they don't move, this means that there is no oxygen, which is going to be what? Which is going to be taken. But they cluster around the algae filament down there on exposure to what? Exposure to light around here. Yeah, now deduction 3. Now this is our deduction. We are saying that darkness prevents photosynthesis, which stop evolution of oxygen, resulting in anaerobic conditions that do not favor aerobic bacteria. So in the dark or in the darkness, photosynthesis does not take place there. So the one we are saying that photosynthesis is prevented in darkness. So which stops evolution of oxygen, resulting in anaerobic conditions that do not favor that do not favor aerobic what? Aerobic bacterial activities. Yeah, then observation number 4, we are saying that there is hardly any object. We are saying that there is hardly any what? Any object, any aerobic bacteria in the ultraviolet region, green and infrared regions of the spectrum. There you can see here. There you can see here in these regions, ultraviolet, there are no bacteria, green, yellow, and the other side of infrared. And our deduction is like light from ultraviolet, green, and infrared regions of the spectrum is hardly absorbed by chlorophyll, hence least absorbed in photosynthesis. So O with no stock little evolution of what? Oxygen. Now when you look at those regions are the ones we marked the other side, X, or no man is what? The one I called no man's land, whereby there most of the chlorophyll which takes place there, most of the light which takes place in those regions are being absorbed by the carotenoids. Therefore, that light does not favor what? Photosynthesis. Yeah, I talked about that earlier. Then there was one of also I'm going to answer the other question Kasim asked me still. Yeah, it was about what? He said that please explain for me more about the mosaic leaf arrangement. Now when we talk about the mosaic leaf arrangement, this refers to the unique pattern of leaf arrangement found in some plants. People, though you can see those branches. An example is that one of coffee arrangement. Look at this arrangement. The one of the jackfruit tree. Actually, the way those leaves are arranged on what? On the branches. What we are meaning by the